So we're going to go over Chapter 15. This is our only day to do Chapter 15 because I assumed it would be a little light here. We won't do a pre-assessment for it today, but there will be a pre-assessment for it next week when we come back and for the next chapter. So just, you guys have the advantage on that because you get to hear me say that when I write it down. So there'll be a pre-assessment for this one and for the next one on Tuesday after the break. Okay, so this one's biotechnology and genomics. So biotechnology, we're going to talk about gel electrophoresis. That's actually what your lab is over, is the electrophoresis stuff and how you can actually use that. It's pretty interesting. That one has more of, like, we think of the relevance of it for, like, criminology and stuff like that. Molecular and reproductive cloning a little bit, and then some of its uses for medicine and agriculture we will talk about. Okay, so biotechnology, there's some start to this stuff. It's always a little easier. But some of the biotechnology that was kind of early on is like the discovery of penicillin. So being able to put a little bit of mold, like this is all like bacteria on a plate, and then they put a little bit of this penicillin, like bread mold, and then there's the whole ring around it showed a kind of a ring of inhibition where we wouldn't grow bacteria. So that's where they started getting this idea of like, what is it putting out there to be an antibiotic, you know, as an antibiotic to kill that bacteria. You know, and then they started using penicillin for everything. So it's like penicillin will cure gonorrhea and everything else. Just started like applying it to everything. Like, it'll work out fine. But that's where we'll get into evolution next week, and we'll talk about why that's maybe not a great idea. So biotechnology, basic definition on this one is that it's the manipulation of organisms and their components to make useful products. We do a lot of like crazy things in science, but the whole idea is that it has a purpose. So everybody forgets because they think that we're doing kind of like wacky, you know, especially when we deal with genetics. Like we must be doing wacky stuff and trying to like ruin the world. I think we're all mad scientists at this point now. But the whole point of it is that we're trying to make useful products. We're trying to solve problems. Sometimes our solutions create more problems, but we've got to experiment them through. We've already kind of been doing biotechnology for years, though, like thousands of years. So microbes to make wine and cheese, using yeast and all kinds of other things to do that is a good example. Day one. Probably. Maybe we'll indulge the holidays. And then for selective breeding, that's been a big one too. So that's some biotechnology is involved in that, of getting like just the variety of dogs and things like that. Domestication of animals. Even selective breeding for like the original plant was a mustard plant, and that's how you get broccoli and cauliflower and Brussels sprouts and all that stuff. That's some biotechnology as well. It's also evolution we can look at. So DNA Technology is a set of modern lab techniques that are used to study and manipulate or make small changes, kind of like manipulate, sort of like make changes by hand. It's like we can move it sort of with our own. I haven't worked all day, so I don't know how to use words today. But so we can work with it, and as scientists, we can make those changes, sort of by our own hands. All right. Genetic engineering involves manipulating genes for practical purposes. So again, when something's genetically engineered, it's for a reason. That's the whole practical purpose, is that it's meant to solve a problem. And the nice part is because there's a lot to these sections, I left all the words on the slide, so you can get these off of Blackboard. Give us something, leave some space for it. Okay. One of the first things that we have to do is kind of some DNA extraction stuff, which now I remember this is one they usually do too, like extracting DNA from strawberries. No, you can just extract DNA from anything, but that's one of the lab parts that we end up doing. Basically, we put cells in a tube or just in some kind of container, and then we use a detergent that kind of messes up that plasma membrane. So a soap is a lot like a plasma membrane, and it can tear it apart. So once we remove the membrane, we treat the cell contents with a protease. Basically just destroys all the protein that's in there. So we remove the cell membrane, we'll remove proteins. We can use an RNase to destroy RNA. So we're just breaking down all the pieces one at a time in there. We'll get rid of some cell debris, so any of the other stuff that we haven't broken down, we can remove those things. And then at the end of it, with the help of some ethanol, we can actually kind of pull out DNA strands in a nice long stringy thing. So that is a lab that we usually do. And honestly, I think you can do it, I think you could do it using household products. So you could potentially do that one at home. It's just not as exciting as doing it like in a beaker in the lab. But that's one thing that you do during that lab. So we have to, if we're going to do anything with that particular DNA, we have to first extract it. So we've got to take it out of the cells. And then we can do stuff with it and process it to figure out all kinds of solutions. So one of these applications that we can use the DNA that we've extracted for is what's called gel electrophoresis. And that's the other one the other part of that lab that we usually do for this. Okay, so it's a method of separating those macromolecules, usually proteins or nucleic acids, for our purposes, nucleic acids. And it'll be on the basis of size. We can also use electrical charge, and then we have some other physical properties. But for the most part, we actually make, it doesn't look very good from like back here, but we'll make one of these ladder-looking situations here. We'll make one of these gels. And it's, like I said, it separates the DNA based on size. I think we'll look at that one here in just a second. It's actually a pretty descriptive term, gel electrophoresis. So cis is sort of like a process. Pore meaning that we've got a gel with some pores in it. So gel with some pores, and then we're going to stick electrodes on it and move something from one side to the other based on electrical charge. So it's a gel that we put electrical charge through, filter stuff through pores, and a process. It is pretty descriptive. It's just a mouthful of a word. Okay. We'll sort of go through this one. The best way, I think, too, is to kind of either do the lab or look at a video that kind of demonstrates what goes on. I'll go through this pretty quickly, but it can be used to separate DNA molecules based on size. So we have a DNA sample put in one end of this gel. A sample is placed at one end. We have a current applied. So the DNA molecules move towards the positive ends from the negative ends. So we'll put the DNA, we'll look at a picture in a second, but we're going to put the DNA next to the negative end, and DNA is negatively charged, so it's going to actually be drawn to the other end of the gel where the positive charge is. So those opposites attracting, it's going to move towards the positive end. Picture works better for this one. Shorter DNA fragments will actually move through the gel faster, and so they travel farther. That's actually a kind of important piece of the gel, is that things that move further away from where we put them in there are going to be shorter pieces. Because they can move through, because they're smaller, they can move through the gel quicker. Bigger pieces will move slower. It will take them longer to get through that gel. So that's an important part. DNA fragments appear as what are called bands. So we can visualize through staining or detecting with radioactivity or fluorescence. So we can kind of light it up and make it glow. That's the fluorescence part. So each band is a collection of DNA molecules that are the same length. Again, if they're traveling the same distance, the idea is that they're the same size. That's a lot of words, but that one's at least on Blackboard so you can look at it. So this is sort of a better visual of what the words are trying to describe. But we have a power source with a negative end towards our sample and a positive end this way. Our DNA sample is going to be negatively charged. It's a negatively charged molecule, so it's going to be attracted towards this positive end. So that's why it's moving this way. So we kind of will load it at the top of the gel. Just don't always look this kind of vertical situation, but that's fine. We're going to load at the top in these little wells. And then we start the gel, or we start the power source, and allow it to kind of be pulled through. Then we get this kind of completed gel. It'll stop at a certain point, we'll turn it off, and it will pull the DNA fragments further down. So those that are far down are the shorter pieces, and these that are up here still towards the top are the bigger pieces. Does that sort of make sense? So that's all I'm talking about. When we load it in, we've got a power source, we turn it on, it'll pull and attract those DNA pieces further down. Once we kind of get our pieces to a certain point, we'll turn it off, and then you can make pictures from there. But the shorter pieces travel further, the longer pieces that are larger are towards the source, still at the top. So at least for DNA profiling, so when it comes to like forensics type stuff and being able to identify things, so it can be identification for criminal justice. It can also probably, let's be real, it could probably be like the whole Maury Povich show. Yeah, one of those things too. I don't know, are there more shows that are also on, like, dependent upon paternity tests? This is the only one I think of. Yeah, so this really helps bring us entertainment. But we've got actually a few things that we can look at to be able to identify the right kind of match of genetics. So there's some repetitive DNA. So it consists of nucleotide sequences that are present in multiple copies in the genome. So we have already repetitive sequences. Like these things happen. And sort of what we talked about is the more sort of repetitive parts that we have, or the kind of extra stuff in there, that if a mutation happens in those areas, that's cool. We'll kind of need some extra areas where if we're going to make mistakes, it won't affect our kind of DNA sequence or expression as a whole. So that's why some of those things are there. We'll also have kind of over time, as far as evolution goes, we have a whole set of what are called STRs, or short tandem repeats. So they're these short nucleotide sequences that are repeated in tandem, so like next to each other, composed of different numbers of repeating units and individuals. So those are the things that get used for DNA profiling as far as criminal justice is concerned, is the short tandem repeats. Because they're going to be a specific size, and then they just get repeated over and over again. So you can use those little segments. So the short tandem repeat analysis will compare the lengths of those sequences at specific sites in the genome. And then we typically analyze 13 sites scattered. So the whole point is that we need different spots. So just in case one of them happens to be the same size, we need a whole set of data on that individual to get a complete picture as to whether, you know, they've committed the crime or that's the person of interest or whatever it is. So it's better to have more kind of specific sites than not enough. The big thing there is the short tandem repeats of the things that are for DNA profiling. So as I said, we've got like 13 sites of these short tandem repeats. But let's say if we've got the crime scene, they've taken the STRs from 2 sites, for example. Like from the first one, if we had the suspect's DNA, when we kind of extracted it, it looked like it was the same size. So if we just went based on that, we'd be like, oh yeah, that's the criminal. But based on a second site, the number of repeats don't actually match. So that would tell us that that person isn't a match. So that's why we need multiple sites to be able to get a really clear picture, because the only one that it can match at all, like 13 sites, is that person. So, and sort of what that looks like is, we'll talk about it, but you kind of amplify the DNA. So you make loads of copies of it, and then they'll put it through the gel the same way that we looked at before, towards that positive electrode. And so our longer DNA or shorter DNA sequences move towards the bottom. Longer ones stay up. But if we're actually able to visualize it, at the amplified from the crime scene, we get a banding pattern that looks like this for those 2 sites. And when we actually look at the criminal or that criminal, but the suspect at that particular, for those particular short tandem repeats, we find that they don't match. The idea that we're looking for is that every line should match. Like it would be here and whatever. Like they should all match exactly. When you look at them, those letters should be the same. But because they're not, even at one spot, it tells us this isn't the right person. Does that make sense? Let's see. Let's see if I can find it real quick. DNA profiling. Like sort of this would be a good example. Really? Of course it would. It's a great example, so we can't use it. Make this larger. Okay. So sort of like what it looks like, because they're never clear. But the idea is that if this is what our crime scene is, these are all our kind of like 13 short tandem repeats that they make this sort of banding pattern. So when you amplify the same things, we can look at each of these suspects and say, well, this one doesn't line up in all the same spots. Even though there's some areas where they do kind of seem to line up, they don't line up all the way through. Because they really, each of those kind of sequences is unique to that person. So that's not going to work. Suspect 3 doesn't quite work either, but when you look at suspect 2, you can see the banding patterns on both of these line up exactly. Because the only way that we can get the exact same banding pattern is if it comes from the same person. Okay. So that's how that works. I like to make it. Okay, so how we get amplified DNA? So amplified just means that we're going to get lots of copies of it. Okay, because that's the cool thing is that we could get like one tiny sample. That's why if like a hair or like one drop of blood or whatever is left at a crime scene, we can take that DNA and make so many copies of it. We can make massive amounts of copies of that DNA. That part's not the problem. So the way that we make lots of copies of that DNA is through a thing called PCR, or polymerase chain reaction. So it's a technique where we take a specific segment of the DNA and then we quickly just make tons of copies of it. Like we don't physically do it, you just kind of put it all in a machine. The machine does it. Like a magical machine. But I used to borrow, when I did research, I had to use somebody else's PCR machine, and it was a really old machine that was like held together with duct tape. Like really? I think they've updated some stuff. But here's sort of kind of the quick overview about how PCR works. Is that there's sort of 3 basic steps. That's not that hard. It's basically what we did with DNA replication, except for done multiple times in a laboratory kind of set setting. So the first reaction is heated, and we separate the strands with maybe something like a helicase. We're just going to separate the strands. So just like we did for DNA replication, we first start with separating the initial strands. Then we cool those strands. Primer molecules hydrogen bond to the target sequences of the DNA. So we heat it up, break it apart, and then we cool it and have kind of a primer set in on both sides. Now that it's sort of stable, a DNA polymerase, so the enzyme that builds a polymer of DNA, will build a new strand by extending primers in the 5' to 3' direction, just like we did before. So we have another DNA polymerase come in and just start building new DNA strands on that target sequence. And then that's it. So we separate it, we put a primer there, and then we put a polymerase in to build those strands. The idea too is we actually have to put in a whole bunch of like nucleotides in there, but we would add those as a scientist. We just throw in a bunch of free nucleotides so we could make that thing happen. Okay. But once we do that, we just repeat it over and over again for like a long time, and then we get massive copies of this. So it's not the greatest diagram, but it still shows you what's going on. So if we start with our kind of target sequence and our sample DNA, first thing is we heat it to separate it. Then we add some primers that tell our polymerase where to start. The polymerase will build a new copy from each strand, and then we've got 2 sort of new copies of DNA. Then we go into the cycle again where they're heated and pulled apart. Then we add a primer, and then we add our DNA polymerase, which builds it. We start the next cycle, pull them apart again. So it pulls all of them apart that are in there. So each time that you do it, you kind of get that magnitude of 2, where it's like 2 to the whatever power. Because by the time we're at cycle 3, it's 2 to the 3rd power. We've already got 8 new strands at cycle 3. So this just gets more and more as we go along. Exponentially more copies each time that we do it. And they'll do tons of cycles of this stuff. That's the idea, is we want lots of copies of the same thing. So once we do all that, we can then put our amplified DNA sequences, because they're all the same, we can put those into a gel, and it should make the gel be easier to see whenever we diagram it. So there's a couple of ways that we can also represent that information, and it's with a southern blot and a northern blot. Here's the funny thing on this one is that the original sort of blotting thing was named after, like, you know, Dr. Southern or whatever. And so then, to be jerks, when they made a new type of blot for it, they named it the northern blot because they're going cardinal directions. So the southern blot is actually named after a man, and the northern blot is them just making fun of the man whose last name is Southern. Like, you guys are jerks. But I think they even made like a western blot after that, and there's maybe an eastern one. Like, they just want all the cardinal directions with it. But anyways, what happens with the southern blot, the original one, is that we have electrophoresis used to separate those DNA fragments by size, and then they kind of appear as like a smear, and kind of like smear across this plate. And then we'll kind of put our whole gel into a buffer, and you can stick some paper towels on it, which is kind of fun. But what we'll have is like a nylon membrane, and it'll be coated with certain probes that actually have a complementary DNA sequence to what stuff we're looking for. So we can take this whole smear of crap. We've got really targeted things that we're looking for on this little nylon soaked sheet, and we put it onto our gel. And then when we remove it, it'll give us this nice pretty blot, like when it works properly. I don't know. I don't think I ever had very good luck with those. But if you do them in the future, I hope you have better luck with them. So those are blots, just a way to represent those. Next thing is gene cloning, where we can lead to the production of multiple identical copies of a gene-carrying piece of DNA in a tube from one single molecule. Gene cloning is really just the idea that it's like we're going to replicate the DNA in a lab setting. And that's the easy way to kind of put that. We can actually make our own recombinant DNA. So we talked about crossing over, and meiosis being a way is getting recombinant DNA is to kind of switch some things around. But we as scientists can actually kind of switch around the DNA also. So we can take recombinant DNA by joining nucleotide sequences from two different sources, which are usually different species. This is what's going to lead us into the kind of the GMO stuff. Okay, so one source contains the gene that we want cloned, that we want to make copies of. The other source is a carrier of that gene that we're looking to make copies of, and so we call that a vector. Anytime something carries something else, we call that a vector. So like when mosquitoes carry malaria, we say they vector that disease. So something that just carries or holds something else is our vector. So plasmids are actually small circular pieces of DNA that replicate separately from kind of the whole circular piece of DNA in a bacterium. So sort of like a bacteria might look like is this, and then you've got this like piece of circular DNA that kind of like folds over itself in just the middle. But they have these weird plasmids, which are just circular pieces of DNA that are somewhere else within the little cell. So that's the stuff that we usually target because it's a really small piece that you can deal with pretty easily. And it replicates separately from everything else, so we can kind of target it specifically. So for a lot of this stuff, we're going to end up using bacteria. E. coli gets used a lot, like a lot. So I've worked in labs where they worked with E. coli. Even the harmful kind of E. coli has a really distinct smell to it. And the scary part is when you go into like non-lab settings and you can smell that smell and you're like, oh God, things smell like E. coli, I'm scared. That's happened before. I think it's been enough years that I can't remember what it smells like, so I think that's a little bit better for me, so I don't panic when I go places. Like, why does this smell terrible? Okay, so a little animation on this one to maybe explain a little bit better.
To recombine and copy genes. A restriction enzyme is used to cut open a plasmid, a small circular DNA molecule obtained from a bacterium. The plasmid serves as a cloning vector. The restriction enzyme cuts only at a certain DNA base sequence, called a restriction site. The same enzyme is used to cut up DNA obtained from a human cell. The human gene must be cloned to produce copies for use or study. A bacterium is induced to take up the recombinant plasmid from the surrounding solution in a process called transformation. This isn't as clear-cut as it sounds. Some bacteria do not take up the plasmid, and some take up plasmids whose LAC-Z genes lack the inserted human DNA. A bacterium is placed on a growth medium. It replicates the plasmid at the same time as its own DNA. Its descendants form a clone of bacteria that all have copies of the recombinant plasmid. The ampicillin in the medium limits growth only to plasmid-containing cells. But which cells have plasmids with a human gene? Bacteria without human genes have intact LAC-Z genes. This allows them to break down a substance in the medium that stains their colonies blue. Thus, the bacteria with human genes are in the colorless colonies.
Got a little bit more detail that I think we would go into on that one, but that's sort of the idea about how that thing works. So there's a lot to that one, so we'll go sort of in steps here. Just to show what's sort of happening is that we'll have the bacterium, like I said, usually E. coli, and it's got a plasmid that we isolate. We actually isolate the cell's DNA with that target kind of gene that we're looking for. Then you use the same, what's called a restriction enzyme. Do I have it listed there? No, it doesn't. Okay, so what it uses is a restriction enzyme. And I'll show you this one in a second, but restriction enzyme is sort of like, I don't know, DNA scissors. They're like DNA scissors. So we look for a particular sequence, and we're looking for the same sequence in our plasmid and the gene of interest. So we look for the same sort of sequence, and it will go through with its little DNA scissors and just clip our little DNA strand. So that's what I think of them like, little DNA scissors. And then we've got a targeted fragment that we actually combine with So we take our fragment and we join it with that plasmid again. So we're taking those and putting them together. But you have to think about, we're using a lot of things that we can't actually really see, and you can't put them in a microscope and put them together that easily. So it gets a little hairy on putting all those things together. Like, I'm not particularly skilled at it, but somebody else is. Okay, but we'll put those together, and then we get a DNA ligase, which we had DNA ligase before in our DNA replication part. DNA ligase is sort of like it's like a DNA glue. Ligase will be our glue. So it's basically like a really, you can't see it going on, but it's like a tiny little craft project. We're going to cut the right sequences, and then we're going to glue them back together. That's the idea. So we get the gene of interest into this plasmid. And the cool thing to think about is that we're using probably a human gene, which is from a eukaryotic multicellular thing, and we're going to insert it into the genome of like a little like prokaryotic unicellular thing. But that's the cool part about DNA, is that it's universal in that sense that all of these organisms are using the same thing, so we can actually move them around. Now, whether we should or shouldn't is a whole ethical debate on that one, but that's why why we do it anyway. Okay, so we've got our recombinant DNA. You end up putting it sort of back in the bacterium that it came from. And then we know bacteria grow by binary fission. So they'll just go through and replicate all their DNA and make exact copies over and over again, just by using their normal methods. And then we're able to get a whole slew of bacteria that have our gene of interest. And sort of as it said in the little video, like you can actually determine it based on putting a growth media in there that will stain certain things, so that's helpful for us too. Okay. But we can take those certain colonies and harvest certain proteins, and then also get the genes to be inserted somewhere else. So lots of options based on that. All right, here's our restriction enzymes. Little DNA
restriction enzymes are enzymes isolated from bacteria that recognize specific sequences in DNA and then cut the DNA to produce fragments called restriction fragments. Different restriction enzymes recognize and cut different sequences of DNA. This restriction enzyme recognizes the DNA sequence GAATTC. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence AATT. This restriction enzyme recognizes the DNA sequence CTGCAG. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence ACGT. This restriction enzyme recognizes the DNA sequence CCCGGG. It cuts the DNA strands between the bases C and G. The resulting DNA fragments have no single-stranded sticky ends.
So basically, they're just little DNA scissors. But the thing I did forget to mention is that it does produce sticky ends sometimes. So that's how we're able to recombine it, because that's the idea, is that DNA is not particularly stable in a single strand. So when we have it sort of stuck out, like a single strand part of it stuck out, it gets really anxious to find something to stick to it. So if you use the same restriction enzyme to produce the same types of sticky ends, then they stick together pretty easily. So that's also, we can make that happen. The DNA ligase still comes in and glues it and makes sure that it's all like connected properly, but nonetheless, it's part of that restriction enzyme and making these sticky ends that make that happen. Okay. A couple of examples. This one's mainly for like example purposes. Because we also use, sometimes it'll use yeast. Yeast is a little trickier to use, but sometimes we can use that one for making certain things. So we can do recombinant, we can make all kinds of products that we're looking for. Like we'll use insulin or growth hormone, or epidermal growth factors. So there's plenty of things, a bovine growth hormone as well. All of these things are made through that kind of process of putting them into E. coli. So then we can use them for what we need them for. So that's where that's the idea of like messing with genetics. It gets a little like, is this ethical? But it's always for a purpose. It's not just to make like Frankenstein's monsters. Like there is an answer to this, is that we're trying to make sure that there's more insulin provided for diabetics and things of that nature. So that's that technology being used for those purposes. In here, we've got a couple that will actually use yeast cells because it's a little easier to use. But sometimes we can't use other types of cells. We would need mammalian cells to really have all of the pieces to manufacture what we need. So we would use for erythropoietin, which helps us make like red blood cells. That one has to go through mammalian cells. The plasminogen activators will also have to go through there as well. So a couple of things, like we'll have to use recombinant DNA stuff. It just depends on if we can get it out with a simple bacterium or whether we need more machinery of a mammalian cell. But there's just some examples. So recombinant cells in organisms can mass produce gene products. So we're exploring mass production of gene products for whole animals and plants. One I know of is like an example where they're trying to use goats and be able to get insulin to be produced through the goat's milk. You know, I don't know if it works or not, but that's the idea, is to get more insulin mass produced through like goat milk. We have recombinant animals, but they're difficult and costly to produce, so there's a little bit of like cost effectiveness on that. But we can clone to produce more animals with the same traits. That one gets a little bit tricky too. So that's, I think, what that was referring to. There's the goat that we're trying to produce insulin through its milk, if I remember that one correctly. As far as cloned animals, we had the first sheep clone, Dolly, back in the day. She didn't live very long. I have to think about the idea that the cells were already at a certain age, and we have those kind of telomeres, so the ends of the chromosomes will shorten over time, and that leads to the aging process. So those telomeres had already shortened when they cloned her. So she was already sort of like aged at the time that she was cloned. So she didn't live all that long, but, you know, I guess there's the idea, at least you could do it. There's a purpose to it, there's just not. I don't know that one to be able to speak on that one. I'd have to do a little bit more research on Dolly for that. So DNA technology has changed the pharmaceutical industry, because with DNA technology, with gene cloning, we're able to produce medicines and diagnose diseases. There's a whole series of like gene therapy stuff that I don't think we talk about here, but some of the gene therapy stuff and solving some kinds of genetic disorders come from this. Therapeutic hormones, too, are produced by DNA technology, like insulin, again, human growth hormone for helping to treat dwarfism. The tissue trace plasminogen activators can help dissolve blood clots, reduce risk factors for heart attacks. I'll get words out eventually. If they're on the screen, if I can't get them out. Yes, there is like sort of just like anything else, there's like different strains of it. I don't want to say different species because the species is E. coli, but there are different strains. So there is the one that's in foodborne stuff that is particularly dangerous, but the strains that they have created with this stuff are usually not harmful. Like if you were to eat the lab E. coli, I don't think that it would produce the same sort of like effects. Does that make sense? So it's sort of like a non-harmful version of E. coli, but they use them in different ways so that you don't ingest it from that. We're just getting the target genes out of it. Yeah. But it does sound alarming, like when you think through it, like this is the stuff that makes people like throw up and almost die, and we're using it for this stuff. But yeah. Does this sort of make sense then? Okay. I'm like napped today. I haven't had all that much caffeine, so I don't even know if I'm saying the right thing. Just yell at me if I'm not saying the right stuff. If I'm not using my words properly. You guys look tired too. What can I make you think? Okay. You guys came. So DNA technology is used to test for inherited diseases too. So that's another one that we could look for. So some of those inherited diseases are like Huntington's disease. Cystic fibrosis is a big one. So looking at sort of like lungs filling up and all that kind of stuff. It's inherited stuff, so we can look through it through DNA technology. Detect infectious agents like HIV. So we can detect for some of those. We can produce vaccines. Really just derivatives of those harmful pathogens that help us stimulate our kind of innate, acquired immune defenses, so preventing diseases. I was so glad this year because we've had more kids in our building, and so it was so nice that I got my flu shot, because I was right at the 3-week mark when it was like all these kids that were near me at certain points had the flu, and I was like, yes. Though with my luck, they would have some different strain, so I still tried to keep away from them and wash my hands because they're gross. They don't. Oh, it's so bad. I'm gonna end up doing it. I probably have to do it next semester, but I end up doing like this whole like germ lab. Like I fed it in just to show the kids how gross they are. Sometimes it helps. The last one was a bottle of insulin, and this is just sort of the production stuff for DNA technology. Okay, here's the other application. Like, it's talked about a lot, but I don't know that there's all that much information about it, so we can talk about it. But since ancient times, people have selectively bred for stuff. And this isn't exactly the same as GMOs, but either way, it's a good example is that the genetics of certain organisms are not the same as what they used to be. Back in the day, corn was like this tall. Now, if you walk through cornfields, they're this tall. That's selectively breeding for taller and taller corn plants. So the genetics now are not the same that they came from of the like native original plants. So that's sort of changing in genetics over time. DNA technology is quickly replacing traditional breeding programs to improve the productivity of agriculturally important plants and animals. So sort of the consumer issue is what sort of drives some of this stuff. It's for a purpose. So genetically modified organisms contain one or more genes introduced by artificial means. I don't know that that's the best definition for that, because in my eyes that means just about anything is genetically modified because it doesn't have the same genetics that it had before. But in speaking with other scientists on this one, when we say GMO, we're usually referring to transgenic stuff, which things that contain at least one gene from other species. So when we say we introduce genes by artificial means, it's usually transgenic. I guess the thing is that's different on that one is like technically you introduce like DNA into your body. Like if I eat an apple, the apple does have DNA. I've introduced genetics into my own body. But it's not the same. It's sort of a natural means, if you will. So this is saying we're doing it artificially and we're adding different organisms sort of together. I don't remember how much I have on this one. Just a little, like, what's the whole point about solving some of these problems? So GMO crops may be able to help a great many of hungry people by improving food production. We can get more out of it. Potential shelf life. Pest resistance is a big one because, again, it's partly the consumer's issues is that when we go to the grocery store, we like to buy produce that looks nice. Like, doesn't have, like, little holes in it or blemishes, like we want it to look nice. And so part of that is that there's been genetic modification of certain plants so they can resist insect kind of chewing on them, which is what ends up happening. Some of the ones I know of that have been fairly successful are the BT, like corn, cotton, soy. I think those I have used BT. And so it's basically just introducing a bacterium into it that goes into the whole plant, and actually when like a little caterpillar tries to eat the plant, the caterpillar dies. But it doesn't have any effect on a human body. That part's been studied pretty well. And I would say that I know about the BT stuff because when I was doing my own research, there's the guy next to me who was doing research on BT. I would say he was very attractive, so I learned a lot about BT. Because I'm like, I'll listen to you talk about your research.
And there's still on the vegetable is to see, say, organic meat, and of course,
Sometimes. And sometimes, so organic is a different set of classifications. Like they have a set of rules that they have to follow, but they're not the same rules, which is interesting for traditional versus like organic. Traditional agricultural actually sets limits on how much you can actually spray with a particular pesticide or whatever. For organic, they choose which pesticides that they say, like this one's certified as organic, but they don't typically set limitations on how much they use. So from my understanding, it's kind of a case-by-case basis on that one about which one is actually sort of better than another. And it's the same with GMOs, is like which one is better than another. Like you look at something like golden rice, and this wouldn't be in shelves or in stores at all because it's made for areas where they have like low vitamin A and where they just eat a bunch of rice. So they're giving a rice alternative that actually just has daffodil genes in it. So that way it can have more vitamin A, so it's a healthier thing for the people who eat it. Now, I think the research just says that maybe they haven't eaten it or just maybe it hasn't worked as well as they had hoped, but that's like long-term research. Like this one seems okay. The BT stuff seems okay based on the research that I've heard. But then there's stuff like where they'll make recombinant bovine growth hormone through these like cows. There's not a thing good about that one. Like it just makes the cows miserable. It leads to possible infections in milk. It's just everything about it is terrible. So I think with all of these things, like we have to look at them at a case-by-case basis because like apples, even for pesticides and organic stuff, may be different than like celery or like, you know, oranges. Like everything works a little bit differently. And I personally don't research it enough, so I'm just like, I'll just take the traditional stuff and I'll deal with it. But, you know, that's the only bother that I have with people who are like, oh my God, I only shop organic. It's like, but do you know what all the regulations are? Because I know a little bit about them, which is, I guess, why I just don't care. But I think it's up to the person to figure that one out. Some people are like, I only eat organic because it tastes way better. And I'm like, okay, I don't get that. It's cool that you do though. How about it? I'm also cheap, so I'm just saying if it's cheaper for me to not buy organic, I'll do that too. I always have a concern though. It's like if we get in this idea of like GMOs are expensive and everywhere, it's like, will people just stop buying produce and just be like, well, I'm not sure, so I better get the Cheetos? And it's like, that's not even real food. Like, I don't know, we're so scared of what's going on with like real food that we're losing, like, I don't know, somebody called it food-like products, that people end up moving more towards food-like products. So, like, just explore all of it, leave options open, just eat regular food, it'll be fine. Until it's not, and then I'm sure there will be data at some point, and I'll have to switch up my stance on that, but that's science, you know. Some days you're like, yes. Like back in the day with the whole DDT thing, they were spraying DDT in the air, and kids were eating sandwiches with it. And they're like, but it's murdering all of the birds. Then you have to change your perspective and say that stuff is bad. So data and time will help us figure all these things out, I think. All right, so genetic engineers are now creating plants that make human proteins for medical use. So we can have rice to treat infant diarrhea, corn to treat cystic fibrosis, safflower to treat diabetes, duckweed to treat hepatitis. Very crazy. Those I don't know a whole lot about, but it seems pretty cool. You could just like feed rice. take any either. So although promising, no plant-made drugs intended for human has been approved or sold at this point yet. Those things take a long time to get approval. So scientists use safety measures to guard against production and the release of new pathogens all the time. I visited a friend of mine, and it's like they don't even have anything genetically modified. It's just they want to release a particular insect to control weeds in Florida. And so I visited her lab where they have this stuff. They have an entire quarantine facility, and there are like 3 doors that you have to go into it. Like the security measures on things that are not even genetically modified are pretty intense. So I can't imagine for other research where where they are modified and getting them put out into the production line. I mean, even when I dealt with some that we had personally mutated. It was like they were in a greenhouse and they were separate from the rest of like everything else. If there was going to be an issue, they would just like burn it all before they started throwing things out. So it's I don't know. They think they've kind of put them out and things spread around a whole lot, but there are a lot of safety precautions in place. We try to guard against rogue microbes. We set lots of guidelines. Tons of guidelines are set with strict laboratory safety, just like the quarantine facility I'm talking about. The genetic crippling of transgenic organisms to ensure they cannot survive outside the laboratory. So that way, if they were moved out, they would just kind of crap out on their own, but they couldn't actually persist in nature. And then for prohibition on certain obviously dangerous experiments, like you can't, like we're just not going to let people just do whatever they want. Like we've got to have confines and rules and regulations and permits to do certain things. So most of the public concerns centers around GMOs being safe for humans and the environment. And like I said, I don't think there's a clear-cut answer on that one yet. Which is fine. I know. So we can have opinions either way on it. And none of it bothers me. It's just, from what I see, there's just not enough data yet. It also comes because I have my students I make them research a GMO every year, so I've seen different GMOs and I've seen research, and I'm like, some of these are really okay, and some of these are just not, for lots of purposes. Now, a common GMO, if you didn't notice, is the glowfish. Have you seen them in the pet stores, the little fish that glow? Those are a genetically modified organism. They've got like green fluorescent protein from, I think, like a zebrafish or something in there. Or maybe it's from a jellyfish. But it was supposed to be an environmental thing, and then people thought it was cute and put it in fish tanks. So here you go. All right, so gene therapy, the other kind of thing. I'll just briefly go over this because I don't have a whole lot for this one. But alteration of diseased individual, it's hard for words today, a diseased individual's genes for therapeutic purposes. So one of the possible procedures, we take a gene from the healthy person, we clone it, convert it to an RNA version, and then insert that RNA genome into a harmless virus. Maybe bone marrow cells taken from a patient are infected with that particular virus that's got the DNA or that RNA in it. The virus will then insert the DNA version into the genome, including the normal human genome and the cell's DNA. And then the engineered cells are then injected back into the patient. The kind of crazy thing about that is going RNA back to the DNA means that we're using a retrovirus, which the only thing that I know of that has the retrovirus would be HIV. So it's sort of be using like HIV as a means of actually doing gene therapy. Now I might have that one wrong, but that's the idea that we're looking at on that one. So I don't want to take up too much time, but that's at least sort of where that one's at. Okay, last thing I think on this one is mapping genomics. So they tried recently, and I think they're still working on this one. There's a whole human genomics project. It's a whole idea of figuring out what are the sequence of like A's, T's, C's, G's, all those things, and all where the genes fit and where everything lives in all of humans. Like they've mapped out the genome for a couple of things. That's why in genetics classes they use like Drosophila, little fruit flies, Because those little guys, I think, only have like 4 chromosomes. So they've mapped out like where the red eyes are, where its little crazy antenna are, where its little wings are. Like they've mapped all those things out. So they know where those genes live. They're still working on figuring that stuff out for humans because it's not always very clear-cut either. We've got polygenic stuff. We've got all kinds of stuff happening. So we're trying to map genomics. We need to define it, and then we need to describe the genetic and physical maps. And for the most part, I think this one's just kind of definition-based for you. Is that really all I want you to know is the basic kind of definitions of what we have with genomics. So the genomics is the study of the entire genome. So that's all the genes, all the DNA, all the sequences, where those things are located within a whole organism, that species. Genome mapping is that process of finding and locating the genes on each chromosome. So where do those genes live at? A little easier to do with little tiny fruit flies. The fruit fly sort of the plant world is this thing called like Arabidopsis, like mouse ear something or another. But it's just a little ugly weed looking thing. But they've mapped it out, so it gets used all the time for genetics too. A genetic map is an illustration that lists the genes and their locations on a chromosome. I think we saw that when we looked, last week we looked at kind of linkage maps about how those different genes are linked together and maybe on the same chromosome. So a linkage map gets us closer to this kind of genetic map, just kind of showing where each of those things are. Genetic markers. So this is a gene or a sequence on a chromosome that segregates or shows genetic linkage with a specific trait. So if we see those particular genetic markers, it's saying this is, we have this genetic marker, this particular gene usually follows it. What else was I going to say? Let me add to this real quick. Just the whole genetic map, just so we have it. We're including with that, we're talking about linkage maps. And don't forget the idea of like crossing over. And those kind of recombinant frequencies were what were included in that one. So I didn't want to forget that. Yeah, look back at the section on crossing over and the linkage maps for kind of connecting that. The physical map is a representation of the physical distance and nucleotides between genes and genetic markers. So there's a lot of things that have to go on as like looking at how many nucleotides are in between, where those things located, what's associated with that particular space. There's a lot. And it gets complicated when you've got an organism with 23 pairs. I suppose if you're not, you've got the video. Lastly, so this is sort of the idea of what one might look like. The idea is that we've got like the centromeres here so we can see what's going on. Each of these has their own kind of banding pattern and they'd be associated with different genes at these different sites. You could sort of look at distances or what things are represented there, what it all means. So it's where you see those banding patterns on those particular chromosomes, that's kind of what that part's referring to. That's all I have on that. There you go. Not super exciting, but it gets the job done. Okay, so for I guess the recording and whatever else is that, because the last semester this particular experiment did not get set up well. So I actually do have lots of resources that can help you look at it. I mean, that's the other part is you could go through YouTube or whatever else. So everybody, if you're watching this whenever, and you guys are still responsible for that particular lab, however you decide to do it, but I'll give you our resources on how DNA is extracted so you can see that process. I think usually it's done with strawberries. And then we'll also look at how gels are run with dyes. And if the ones that I have for you don't like seem to work as well for you, there's loads of YouTube videos that you can use to find kind of something to help figure that one out.